Hi, this is Miss Litton, and this is my wonderful Period 3 class. Say hi. Hi. And we are continuing on with the immune system. Before I continue on, I want to backtrack just a little bit so we can think about it, okay? So the immune system is talking about what in our body? Protecting, defending our body. So what are some ways, some barriers we have so we don't even let anything in our body? So let's name some things. Yes. Skin. Oil. What else? Just say some. Hairs. What else? Skin, oil glands, hair. Is that the only thing we have to keep things out of our body? Yeah, you're there. Say it. What in the stomach? Low pH. What else could we use? Ciliated cells will keep things out of our body, right? So those are all things that keep things out. Now, let's say they're able to cross that barrier and get in. Which immune response will come first, innate or adaptive? Innate, what we're built with, right? So let's think about some of the cells we're built with to initially handle the problem. There was a whole group of them called, started with a G, granulocytes, remember that? Okay, and all of them, regardless, most of them like to do what to anything that comes in our body? Eat it. They like to eat it. Now, once they eat it, there are different strategies on it. They're the first on the scene. Who was the first on the scene? That was a granulocyte that was first on the scene. Most numerous. Neutrophils. They come in. Okay? Do they have any memory if they've seen that disease before? Yes. No. Your innate response has no memory. It cannot have an amplified response if it's seen it before. Every single time, it's something new to them. Okay, all they know is this doesn't belong here, so I'm going to attack it, eat it, or destroy it. So when the neutrophils get on the scene, they're going to eat. Then something a little bit slower to the scene, but it's more long-lasting as far as consuming and eating things. Do you remember what that was called? It starts out as a monocyte. Yes, and then it becomes a macrophage. And the macrophage is more long-lasting. It's going to eat. And these cells will eat until they die. And when they die, they become categorized as what? Pus. Exactly. Those are dead white blood cells. You have other cells of your innate immune response. One of them releases a chemical to make your blood vessels leaky. So it makes it easier. What is that? Histamine, and what's the name of the cell that releases it? Mast cells. Mast cells out in your tissues, there's another name for the one that's in your blood. Starts with a B. Basophils. Okay. So basophils are in your blood, mast cells in your tissues, they release histamine. Histamine makes your blood vessels leaky. Let's talk about some consequences of leaky blood vessels. One consequence is cells can get out more readily to fight at that infection site. What are some other consequences of having leaky blood vessels? Yes? Swelling. Good. Because what pressure is greatest at the arterial end of a capillary bed? Hydrostatic. So it's going to force more fluid out. And what's supposed to recover it? Your what? Oh, well, osmotic pressure will bring it back in, but what recovers the excess tissue fluid that's out there? Lymphatic system, right? Which is good because it's going to have to pass through a lymph node, so you can check to see if there's any harmful pathogens in there. But in that local area, we're not going to recover it as fast. It's going to cause swelling. What's another consequence of this? This is called an inflammatory reaction. What's another consequence of an inflammatory reaction? Do you have your notes in front of you? Could you be, what? Localized increase in temperature. That could be a very good consequence because it could get warm enough to kill off the bacteria. What's another consequence? Yeah. You could have pain. Yeah, you could have pain because the swelling pushes on the free nerve endings and that can hurt. That also sends off an alarm to you if you have pain in that area that there's something wrong. So that's your local response. That's your innate response to that problem. Now, we then have our adaptive or acquired immunity. This immunity gets better every time we use it, okay? And there are two strategies, two different responses to deal with the problem. Because viruses or bacteria, they could be free-floating in your tissue fluid. They could be in your blood, being independent. And then you have some viruses that could go what? Inside of your own cells and turn them against you. and become factories to produce even more viruses. So we have two different strategies to handle the problem. Strategy one are the B cells. Okay? B cells have to be with the antigen directly. They come in contact, their target is the actual virus or the bacterium. It has to come in contact with it. There are B cell receptors that will come in contact with that particular antigen. Once they come in contact with it, B cells will grow and mature and produce two types of cells. What are the two types of cells they will generate? Plasma cells and memory cells. The memory cells are for next time, to respond more rapidly. What will the plasma cells generate? Antibodies that will be in your plasma, right? Right? They will be in your plasma. So when you think about B cells, do B cells do any killing themselves? No, they don't do any killing themselves. They come in contact with the actual pathogen. They're stimulated to grow and produce antibodies. And through clonal selection, you will have a clone army of B cells that all produce antibodies. These antibodies will tag the pathogen by binding onto the antigens. Antibodies have two arms, so they can hang on to two antigens at once. Okay? And they will then, the base of the Y is what determines how this thing will be destroyed. The B cells aren't actually doing any destruction themselves, but they're lending it, so when it is marked with an antibody, it makes it easier, for a macrophage to come along and engulf it and take it out. Okay, or some other process to kill it. So that's strategy one. Direct contact, free floaters, viruses, bacteria cruising around in your body. Strategy two are the T cells. The T cells, they take care of our own body cells that have been infected, that have a virus inside of them. Remember, our bodies constantly display, our individual cells of our bodies constantly display what's inside of them. Constantly display that. Blue tells Slate, why is that a good adaptation for our body cells to constantly display what's inside of them? Go, Blue.
Yeah, I know that that's happening. Then they can respond. It's a quiz.
Okay, why is that a good adaptation to display what's inside of you? Why is that a good adaptation? Come on now, speak. Yes?
So if you have a virus that took over, you can show and do apoptosis.
Yeah, right. Because you can't, there's that cell membrane, that barrier, right? And this is an easy way to monitor what's on in our cells. What won't get displayed? Do you have any idea what won't get displayed? Think retroviruses. Yeah, if it has integrated into our own DNA, we can't display it because we don't take pieces of our DNA and display it. So we can only display what's available in our cytoplasm. We can display that. Okay, but it can't display something that's embedded in our DNA. That's why they can remain lethal for, remain latent as a provirus, right? For years and years and years because we can't display it. And then something could trigger it to come out. Okay? So the T cells, when you want a T cell to talk to, and this part we didn't get to last time, T cells, the only way you can talk to a T cell is when it's displaying something on either an MHC1, to even kind of introduce it a little bit, or an MHC2. Yeah, you could have this particular virus could be crawling all over this a T cell that would recognize it. It could be crawling all over it and going, no, 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 and the T cell will go, no, 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 I can't hear you. The only way they know about something is if it's displayed on an MHC1 or an MHC2. That's the only way you can trigger it. Once it has been triggered, okay, so this is a body cell who's going to display this antigen on an MHC1. Now this T cell will do cell-to-cell combat to wipe it out. If this were a dendritic cell, if this were part of your innate immune response presenting it, it wouldn't present it on an MHC1, it would present it on an MHC what? 2. And at that point, you're going to help it, and you're going to facilitate whatever it's doing. You could release cytokines to amp up the immune system. Okay, so that's how you talk to T cells. Not it. Whoever is not it, guys, stop. Whoever is not it, decide which one you want to be. Do you want to be the B cell or the T cell? Go.
B cell, what it is, that looks for, so in the bone marrow, and then they're chilling with blood when they see a bacterium. They come and attach to one with receptors, and then from there, treating antibodies. One of them is a plasma cell, and the plasma cells are what goes into the brain. And it has memory cells.
Oh yeah.
Cells they attach to it. Antibodies attach to it.
Which box are we in, first of all? Oh, okay, perfect. Okay. All right. Now, remember back when I presented this last time, right? So you eat this dendritic cell, which is part of your innate response, is recognizing self from non-self. So it takes anybody that's non-self and he eats it. And then he displays. Now, when he displays it to this helper T cell, from what I've already talked to you about, would it display it on an MHC1 or an MHC2? MHC2. MHC2, I will help you. Okay? You're displaying on an MHC2, so it's showing it. It's going to display it to this T cell on an MHC2, and I've got slides and stuff. I'm going to teach that to you. I'm just asking if you know it already. So it's displaying it inappropriately to this helper T cell. Now this helper T cell is activated. Once he's activated, we can see here this B cell. Notice he comes in direct contact with the pathogen. The T cell did not, but the B cells do. Okay, and the B cells make something that's going to come in direct contact with the pathogen, right? And if he then is connected to that pathogen and a helper T cell confirms it, and this is for a virgin response. This is for you've never been exposed to it before, okay? Then it will divide and make two types of cells, plasma cells and memory cells. And remember, the memory cells are for next time. The plasma cells will begin secreting antibodies. These antibodies are specific for only that pathogen, no other pathogen. And then here, that antibody marks it, then some other cell will actually destroy it. If this was a non-virgin cell and you're dealing with just a memory cell right here, a memory cell does not need the conformation with the T cell. It doesn't even need to be exposed to the whole pathogen. There's the part on the, and it's not in your book this way, but I'm just explaining it to you. On the antigen, there's a region of the antigen called the antigenic determinant that actually binds with the B cell receptor. And that antigenic determinant is what it docks into to know that it's bad. But when you have a non-virgin, you could have just a portion of that antigen. You could have one that's been partially digested and just a little, it's called a Hampton, it's just a little piece of the antigenic determinant, and that is all it needs to trigger and start to generate its antibodies. It doesn't need all of the other confirmation that we do before we develop these clone armies. It's like, I know it, I know what it is, it responds rapidly, and that's why you don't get sick the second exposure. Now, why would somebody get sick 5 times with chickenpox? Yeah, different strains. And this is when B cells are like, I don't recognize you, and it goes through the whole virginal response again. And so that's why they get sick and sick and sick. Okay? All right. Then when we finish talking about the B cells, did I show you this one at all? Yeah. About blood types? But I don't think I asked you this question, did I? No. Okay. You are yourself. And you guys, I have this AP bulletin for you. Have you got one yet? Okay, so you can pass these around. I don't think they gave me enough. If you didn't get one, I'm sure there's one at the student store. If I don't know. Tell your bio buddy, do you have a College Board account set up? Yes. That's what I like to hear. Remember, if you're getting ready just for your straight up SATs, they'll send you like a word of the day, things like that to help you get ready. They are fun.
You have to take it.
And then
You have to take it one.
you're all going to sign up for the SAT 2 in biology, yes? Yes. Oh yeah, we had this discussion. Did everybody get one? Did I have enough? Oh, okay, great.
What?
Because I had a couple absentees.
What did you say? This year you're taking what? One or two?
And you have, oh good.
They say one is like no purpose.
There's no. I'm taking two next year.
Because two, like I'm taking it now because math analysis is when you finish chapter 7 and 8. That's like it. That's everything I need to know.
Really?
Because I talked to Lori about
Because she knows.
Let's go,
Good
peeps.
one, buddy. Right. Is yours working?
wait, you have to pay for this.
Yeah, of course.
It's like, will we be like missing out on the class for these?
For what?
For the exam?
Oh, I know what day that's on. Okay. Yeah, I'm just looking to see what day that is. It's a 2-4-6 day, so no. Yeah. Yeah.
It's like 3 hours.
Max.
Yeah, I just logged in. I'm sorry.
You want to sign up for your SAT too, right? For biology. It only takes like an hour to take it. That test for you guys is going to be like, you're just going to blow. I expect you to do very well. My honors bio students score very high on it. You better score lights out. I don't want you missing any questions on the SAT too. None. 0 questions. Okay? You know I have scheduled a Saturday and one Sunday to take practice tests. I'll ask you right beforehand how many of you are coming just so that I have enough tests ready. There's not a limit. Somebody asked me that in first period, like how do we sign up for it? I just need to know general numbers so I have enough tests ready to go. Usually we spread out into a couple classrooms because I can't fit everybody in here, so I just take over another teacher's classroom. It's pretty chillaxable during that time. I just try to give you enough time so you can experience what it's like taking the test. I have you take the multiple choice part, and as soon as you're done with the multiple choice part, you can take the essay part, and then when you're done, you can look at the rubrics for the essay, and you can look at the answer keys for the multiple choice, so you know exactly, what you would have scored. You can, add it all up so you have a good idea. So there's one, I think the first one's on a Saturday and the second one's on a Sunday, and that will be a different practice test. I have 2 available for you, and then you'll be able to go again. Questions? Yes? Okay, yes?
For the SAT subject test, do we sign up for it on the college board?
Yeah, you sign up for that on the college board. So whereas the AP exams, we administer AP exams here at the high school, and that's given on this campus during your regular school day. The SAT 2s, we don't administer them here at our school. Bigger schools, neighboring schools do. And so the sooner you sign up for it, the closer you'll be taking that test. So, I think they have them in Agora and things like that, so you can sign up, and literally it takes you about an hour or so to do it. And remember, there's the SAT 2, there's an E and an M. The E has an ecology focus, and the M has a molecular focus. You are ready for both tests, so you don't have to worry like, oh, I wonder, and you don't have to sign up for one or the other. And the first part of the test, everybody takes. Did I already tell you all this? The first part of the test, everybody takes. And then you go to the back of it, and there are E questions. Let me just pause this for a minute. Okay, we're back. So let's see how we did on this one. Yay! Michael. All right. So the 2 arms, I want to reiterate the base of the Y, there's only so many ways to die, but the base of the Y tells you how that's going to go down. The arms, okay, that's the highly variable region. And the importance to remember here, because this is one of your 3 systems you've got to know, why is it that we can recognize millions of different pathogens out there in the world? It's all due to what? Exon recombination, right? Post-transcriptional control, right? Once you do transcription, you put on a cap and a tail, right? 7-methylguanosine is your cap, poly-A tail. And then you could have where you cut out some introns, you cut out some exons, and depending on how you combine them, you'll get a different product every single time. So this different products are these different arms. So this is the highly variable region that's going to be exposed to that antigen. And that antigen is attached to a whole organism, right? All through exon recombination. All right, and you can see that here in this picture as well, looking at the way the different genes are combined together to get some variety. 2 arms, the benefit of 2 arms is that you can hang on to 2 antigens simultaneously, which causes clumping and keeps it together so that you can destroy it right there. The base right here is going to dock into a receptor on the macrophage, so he knows, oh, I'm supposed to eat you. And then there are different kinds of antibodies, and the name you want to know is immunoglobulin. Another word, opsonins. You could write that down somewhere. Opsonins, opsonins, opsonins. Okay, and these gamma-globulins, did I show you this picture at all? Yeah. Okay, so the different ones, and the one I want you to really, IgG, immunoglobulin G, is the most common. Ways to remember some other ones if you just wanted to pull an example up somehow. IgA is found in your throat, and the way I remember that is if somebody wanted to look in your throat if you were sick, you would go, say, right, IgA. Okay, the way I remember IgE is the base of it goes into a mast cell. If it's in a mast cell, and a mast cell is going to secrete histamine. Histamine, you're having what sort of response and allergic response you could be having? And I think eat you, and that's how I remember IgE. Okay. And then here you can see, just blue real quickly, listen as Slade explains this. Go ahead, Slade.
Very cool.
All right, so this particular antibody is underneath the classification. There's only 5 classifications. This would be IgE, right? IgE is what will dock into a mast cell. All right, that is the strategy of direct contact, right, with the antibody mediated. Another name for this same system is humoral response. So you could add this in somewhere, is just different texts, you know, call it different things, but that's kind of an old school name, is humoral response for antibody mediated. You could remember, if you know what a humor is, like if you know your writings in your other classes, you've read about the humors, that was the, like the body, the fluid of the body, or the humors. So that's the humoral immune response. Okay, now let's do cell mediated. Keep in mind, the cells you're going to be attacking now are not going to be prokaryotic cells. Okay, they're not going to be viruses. These are going to be other eukaryotic cells. Where's the source of these eukaryotic cells? Either your own body's eukaryotic cells that have gone bad, or if somebody else's cells are in you, right? This is attacking another eukaryotic cell. And so here you can see this is this target cell. These are all cytotoxic T cells that are trying to destroy this target cell in the body. And when you talk, okay, the T cell can only talk to another cell if I was just trying to create 2 small cells there, is if an MHC1 or an MHC2 is displaying that particular antigen. Otherwise, the T cell cannot hear. So MHC1 right here is displaying it, and here's an MHC2 displaying it. What's going to happen to the green cell, the one on the right? Any ideas? Because I know you all watched the Bozeman I asked you to watch. If it's on an MHC1, it's displaying it, it's going to get killed. If you're on an MHC1, you're done. If you're on an MHC2, I will help you. Okay? MHC1 and you're done. You're going to get killed if you display on an MHC1. If you display on an MHC2, I will help you. Do you know what type of MHCs a helper T cell has in your immune system? MHC1. What kind of cells, what kind of virus likes to infect a helper T cell? HIV. So when HIV gets in a helper T cell and it displays it, then that helper T cell gets wiped out. Helper T cells are a type of T cell that coordinate your entire immune response. And they get wiped out. Macrophages. Do you think macrophages have MHC1s or MHC2s? Twos. Why? What are macrophages doing? What are they a part of? What are macrophages a part of? The innate immune response that's going and engulfing things when it comes in your body, right? Okay, they have MHC2s. They have get out of jail free cards. Because if they're displaying their antigen, that's because they're trying to look and act as an antigen presenting cell, just like a dendritic cell, and presenting that antigen to try to find a helper, or try to find a T cell that can recognize it. Here's a bummer. Macrophages engulf the HIV virus, but they don't actually destroy it. And macrophages act as a vector, as a conduit, to take the HIV virus to your brain and cause dementia from HIV, because it's displaying HIV on an MHC2 as if it owns that disease and it's taking care of it, but it really hasn't. It remains undigested within the macrophage. Okay, so that's one way HIV is really tricky, because helper T cells, where you'd rather those just stay alive so that if the more virus, it's there to help coordinate the response. They get destroyed. Macrophages who haven't destroyed them, they get left alone and they spread it to other parts of your body. Okay. So here, Blue, you go ahead and take care of this picture right here. Go ahead, Blue. See if you can recognize the type of T cell up at the top as well. And you know what? I realized something, you guys. Before you do this, I skipped over this slide entirely, which makes what you're trying to do super difficult. So let me go over this picture real quick. Up here at the top, okay, up here at the top, what do you have? A macrophage. What has he done? Yeah, he engulfed a pathogen, right? So now what is he doing? He's displaying it. And he's going to display on an MHC2. And he's going to go look for a T cell. Sorry for skipping the slide. He's going to go look for a T cell that can recognize it. Okay? So virgin T cells have to be triggered by an antigen presenting cell to get the ball rolling. Then this T cell, this virgin T cell, will divide and make 3 types of cells, where the B cells only made 2. What were the 2 cells the B cells made? Plasma cells and memory cells. T cells will make 3. They will also make memory cells, but then they make 2. They make a cytotoxic T cell, and his job is cell-to-cell combat and killing. And he will make helper T cells, which are all about amplifying the immune response. They can secrete interleukins, cytokines to pump up the response, to encourage the B cells to make more antibodies, etc. So you get 3 types of cells. So here on helper T cells, let's just point out a few things. He's very happy, wants to help. So they're the major driving force, the main regulators of the human defense. Their primary task is to activate B cells and what? Killer T cells. Good. The T cells have to be activated when a macrophage or a dendritic cell has eaten an invader. It travels to the nearest what? Lymph node and presents that information, and it'll present it on an MHC what? 2. So it displays the antigen fragment from the invader on its own surface. This is called this, duh. When a receptor of the helper T cell recognizes the antigen, it's going to meet a lot of T cells that don't recognize it. That's why it takes 10 days to 2 weeks to develop your clone armies because you've got to find the right cells, the right versions. Then the T cell is activated. Once it's activated, it divides and produces proteins and activates B and T cells as well as other immune cells. Okay? Your killer T cells, these are assassins. That this is an assassin that only points its rifle at a very specific target. Each assassin of that clone army, you make a clone of millions of these assassins who will wipe out that particular, those particular cells that display that antigen. It could be a virus-infected cell, a cancer cell, a bacterium cell. Okay, now, could I have Blue explain now about the T cells? Go ahead. Also include in your description how this is different from the antibody-mediated response. Go ahead, keep going.
Alright, so you're the antigen. Put it onto the helper T cell, and it produces and the virgin T cell, and it produces a cytotoxic T cell, helper T cell, and a memory.
Okay, let's check your notes. T cells and cell-mediated immunity. It targets our own infected cells displaying the foreign antigen. T cells which originate in the bone marrow and mature in the thymus gland. This is why they're called T cells, because they mature in the thymus gland. Where did they originate from? In your bone marrow. B cells also mature. B cells were also originated in the bone marrow, but they also mature in the bone marrow, hence the name B cells. Okay. So T cells mature in the thymus gland. When a virgin T cell is triggered by a presentation cell, displaying an antigen, it will mature and make 3 types of cells. A cytotoxic T cell that kill any cell displaying a foreign antigen on an MHC1 surface protein. Helper T cells, it's like an army general encouraging the response. And memory T cells for the next time. Clonal selection is the same as B cells. Once a virgin is triggered, makes a clone army. What's the clone army going to do when it's done? Apoptosis, except for the memory cells. Okay, now, Slate, go.
Okay. Infected cell, and C1, which if you prevent it, you have to die. I don't see the difference.
So let's see what kind of things we can recognize here.
One's attached.
Okay, so do you see right here this MHC1? The antigen is what in this picture? The red triangle. See the red triangle? That's the antigen. The green is the MHC1 in this picture. The blue coming here are the T cell, any guesses? Receptors. You know about receptors, right? Yeah. Okay. T cell receptors. This binding is stabilized by a protein that comes out of the T cell, and it's called CD8. Now, one way to remember that is if you're displaying on an MHC1, you're what? Done. So CD8, I will ate you, or he ate you, or something, okay? Now notice it says it's an infected cell. Notice this says T little c cell. What do you think the C stands for? Cytotoxic. Good. Okay, now look at our second scenario. Okay, what do you think this H stands for? Helper, and it's displaying on an MHC2. I will help you. It's stabilized with a CD4. I am for you, not against you. Okay? You get 8, or I am for you, not against you. So it stabilizes that binding. Notice it is put, who is displaying it? An antigen presenting cell. Okay? B cells can present antigens in this way. Macrophages can. Dendritic cells can. Okay, all of those are antigen presenting cells. So on your notes where it says both B and T cells are specific. Oh, I'm sorry, that's what I meant to do. My bad. I'll get it together. Number 4, MHC proteins, 2 types that display antigens. MHC1 on almost, yeah. You know how to do it. You're like, all cells. I even know the tone. Can be killed or displaying foreign antigen as well. MHC2 on APCs, antigen presenting cells. Antigen presenting cells. Macrophages, dendritic cells, and B cells. Both B and T cells are what? Specific. Their receptors can only recognize 1 to 2 antigens. Therefore, we must have millions of varieties of virgin B's and T's prepared for any potential antigen in our lifetime. I mean, think about it. You're prepared for anything you face in your lifetime, no matter where you go. Now, does that mean you won't get sick? No, you will get sick. Why? Yeah, there's all different things out there, and your innate response can only handle so much. You have to have your acquired or your adaptive kick in order to actually get rid of that sickness. This is why when you get a cold, you're probably going to get it for a couple of weeks. Now, if you're starting to get flu-like symptoms early on, your throat is starting to bother you. Remember, what did I tell you that comes from? Did I tell you where that comes from? When your throat starts to hurt?
Inflamed lymph nodes.
Comes from neutrophils. What neutrophils do is they're like, oh, there's some pathogen over there, and they just like, just utter destruction. Because in their mind is, yeah, we're going to kill a few of our own cells off, but it's all good in the end because we won't let that virus get in. So that sore throat is a result of your neutrophils trying to control the situation. So if your throat is starting to get sore, what's something you could do to help the neutrophils out? Okay, what else? With? Yeah, or Listerine. You're going to help try to kill off that bacterial count, right? Try to lower it to assist it. All right, next. Take a look at this diagram right here. And this is our T cells. Stem cell, all the T cells are originate in the bone marrow. Okay, now these T cells are migrating to the what? Thymus gland, hence the name T cells. But if they don't go to the thymus gland, then they're going to become what? Natural killer cells. Okay, these are our posers who are trying to act like, you know, they're great. They still come from the bone marrow, right? They still come from the same place, but they don't have the specificity. They don't learn. They just have a lot of big guns to shoot. So they'll kill these guys that are targeted depending on the Y, right? The base of the Y might call for a macrophage. That base of the Y might call for a natural killer cell, okay? But here, you could have a cytotoxic T cell that kills from your virgin T cell. You could get a helper T cell, and he's going to bind to MHC2s to help them, and then they'll secrete cytokines that amplify the whole immune response. Ask your bio buddy, are you comfortable with this picture? Yay.
Not quite yet.
Olivia is. All right, you are going to ping pong back and forth. Youngest bio buddy, you are going first. When the colors change, then you change what you're going to read. So, youngest bio buddy, go. Understand what you read.
Inflammatory responses, NPC, protective proteins.
Specific is that the antigen levels is one thing. have bleeding.
You can't step on your body and just straight up unless you have defense mechanism.
Okay, got it? I don't think that's it. Okay?
So you
All right.
can't like string it.
If you're gonna grab that, grab it now.
I think you can. So wait, snap that. Hold on.
Screenshot it. And then crop it and then send
Mute
it.
her.
The camera doesn't work when you FaceTime. I got it. Okay.
Okay, you good? All right. Now, talk through these three scenarios with your bio buddy. Could you justify these claims that are made underneath them? Explain that. Explain those claims.
My max team B cells. Why? What do they look for? What do team B cells look for? The antigen.
There's no MHC markers.
Well, they ignore.
Okay, yes. Number 2, yes.
Because if it's processed, the T cells could do something.
All right, first one. Why do T cells ignore this first one? There's nothing in the antigen, so it's going to ignore it. Okay, why do B cells ignore it? Because they don't respond to MHCs, right? They have to respond to the actual pathogen itself. Right? Why does this start a T cell immune response? Yeah, there's an antigen in the MHC. Okay? Now, this isn't drawn to scale. This should be tiny, like in here, right? See these bumps right here? Those are the bumps right here. Got it? Okay? So why do B cells respond to that? What? Yeah, because they need to come in contact with the actual pathogen in order to trigger a B cell. So this is a great slide because it forces you to apply the differences between those two responses. Yes?
What exactly is MHC?
MHC are proteins that extend from our cell membrane. Remember we said we have channels, we have carriers, we have receptors. It's a type of receptor.
So does it only form when the B cells come in contact with an antigen?
No, MHCs are on all of your cells.
Okay.
Your cells will either have an MHC1 on it or an MHC2. What's the most common MHC? One, and they're found on all cells except what? B cells and antigen presenting cells, macrophages, dendritic cells, they have the MHC2s. Because if they display that antigen, they're safe. It doesn't mean you need to kill them. It means this is what we're working on. MHC2, I will help you. Stabilized with the T cell using the CD4. I am for you, not against you. All right. So here's another little cartoon to help you apply your knowledge. Apparently these pathogens are really angry chefs. He has a club. He's clearly drinking liquor. He's biting. So you have an antigen presenting cell. I don't know why he looks like a German soldier. But here you have a specific immune response with your B cells. Who's patting this guy on the back? Helper T cells saying yes. Then this guy, does he actually have to go to the battle scene? No, he just has to what? And he is releasing what? Antibodies, right? So he doesn't actually, if he's in the Air Force, he doesn't have to go and do combat, okay? Right? All right, now let's look what's going on here. Here's some different pathogens. This big ol' honkin' worm, okay? Here's those bad cooks again, and this one looks like a dumpling or I don't know what, it's got a nail. Here's infected body cells. Not a dumpling, like a chicken leg. That's why did I say dumpling? I don't even want to know. I don't even want to look at it anymore. Okay, here's an infected body cell. See the big bad virus inside of it? I'm so glad this is getting recorded. Here, remember I told you that some of your innate immune response, they go for the big ticket item like a worm or a splinter or something. Those are eosinophils, so they're attacking him. Here's the T cell doing cell-to-cell what? Combat. Here's an APC. What's he going to do? Eat that guy and present it. And here's a B cell releasing antibodies. Okay, take a look at this. When you see something like this, you ought to be able to say a few things. Okay? Ping pong back and forth. Each of you say 2 things that you know, that you could explain from this diagram. Go back and forth 2 times. Go.
They become
Stem cell is what makes either platelet or red blood cell, which are 2 main things out of
oh no, it's the
part of the blood. And then
no.
Fluid stem cell, red stem
here's the B lymphoid.
Do you know everybody's function? What's this one? What's the red blood cell? Carry what? On hemoglobin. Okay? Each hemoglobin molecule can carry how much oxygen? 4. How much hemoglobin is in each red blood cell? 250 million molecules. What are platelets in charge of? Blood clotting. Let's look at these 3 granulocytes. What is a neutrophil? What do you know about him? Most numerous, first on the scene, right? What about this guy? Targets bigger things, bigger parasites. What about this guy? Basophil. What does he do? He's like a mast cell that secretes what? Histamine. Mast cell isn't in this picture, that's why I have another picture here in just a minute. Go over here. Lymphoblast, B lymphocytes. What's important about them? They generate antibodies to target the pathogen directly. T lymphocytes do cell-to-cell combat. Natural killer cells could have been a T lymphocyte, but they didn't go to the thymus, and what they do is they what? Kill. Okay, here's another diagram that I annotated, if it would be helpful to you to grab.
Okay, more nothing.
I got it.
Wait, let me snap that.
Everybody got it? Okay, I also made a Venn diagram in case this would be helpful to you. Where Venn diagrams overlap, what is that about?
Similarities.
Good.
Okay, and then you have other pictures in your book that break down non-specific and specific. immune response, you secrete even more. Opsonins, another name for antibodies. Yay, good little video.
I'm scared of studying this chapter.
All right.
I'm scared of studying this.
And then I put a diagram in your notes already. Okay, we'll just do a couple questions and I want to move on.
I'm scared to study for these tests.
Yay! Good job.
60 feet less than that. 60 feet.
Dude, I'm thinking about it, like AP Chem, I don't know.
Not worth it. Not worth
Are
it.
you gonna take it?
I'm definitely.
Yeah, exactly.
Oh, because I'm doing science. I'm gonna be like science.
You pumped yourself.
Okay.
Guys, focus on the question.
Oh, we have questions, dude.
Okay.
Aww. Okay, you didn't even need the picture. Didn't even need the picture. If you're destroying another cell, it must be displaying it on the MHC1. Yeah, because then you're done. All right. So now, take a look at this. This just brings in another level to what we've already learned. If you look at the top, in immunity, you can break it down into innate. You already know that. Here is our innate response. These are policemen over here. Over here, you have your acquired immunity. So you weren't born with it. And that can be broken down into two categories, active and passive. Active is what we have been talking about, where you actually have cells, memory cells that can respond and can generate more cytotoxic T cells and more plasma cells to secrete antibodies. That can happen naturally because you're exposed to that, because somebody sneezed on you, you got sick by somebody else, or you could go and get a what? Shot, a vaccine, a weakened version of it. So you could go get your flu shot, get exposed to what's out there, build up some memory cells so that if you ever got exposed to it, you don't get as sick. Okay, over here, passive, this is not immunity you own. This is borrowed, short-term immunity. Just in that moment, you will not develop memory cells. You will get sick the next time you're exposed to it, right? If it's just passive. And the reason is because you never trigger, because you have the antibodies borrowed, you never then trigger that ability to develop a response to it. If you look here, one way to get it is through breast milk. Okay, that's not an option for you anymore. Okay, but that is as if you become in a relationship or whatever and you start to have kids, regardless of what you feel about the idea, ladies, of breastfeeding, biologically that's a very good choice. Because the whole time you are breastfeeding your child, whatever antibodies are in your body are now passed to your baby, and that's doing what for your baby? Protecting your baby. Also, it's a very good form of nutrition. Okay, so I would strongly recommend that regardless of your feelings about breastfeeding. Artificial antibodies is like, I got bit by a snake, I don't have time to get sick, I don't want to die, so go ahead and give me the antibodies right now to protect myself. So that is an example of passive. Okay. Second thing here is when you're first exposed to your initial response, your virgin B's, your virgin T's, that first response isn't really big. You've already had it in your body usually a while, and you will have a rise in antibodies for sure to help wipe it out. But your second response to it, it's really high and very rapid in response. Here you can see this is when you are exposed to it, and then here's day 30, so this would be like day 14 or 15, you start to have an increase. Here, in this case, as soon as you're exposed to it, your antibodies jump off the roof. So what is that? Is that a B cell responding or is that a T cell responding in this diagram? Read your graph, peoples. Read your graph. It's got to be B cells. Why? Plasma antibody concentration. My children whom I love, okay? You are expected to be able to read a graph. You should have been able to tell me right away, that says antibody, antibody is B cells, okay? I would say the majority of you who are brave enough to say anything said T, okay? So interpret your graph and look at it so you understand. Those of you who have actually prepared for your exam know that I actually put this in as one of your potential questions. And you were really paying attention as I started to talk about this and not checking out. Okay? On cytokines, we've already talked about them already, and you have two categories mainly, those that are on the offense and those that are on the defense. Lou, could you explain why would I categorize one as offensive weapon and one as a defensive weapon? Go ahead.
The offensive would get it before it affects anything. And the interferon will fight it once it's ready. Yeah.
Okay. So why is interferon categorized as a defensive weapon? Who secretes it? Who secretes interferon? Okay. All right. Now, so interferon would be considered a defensive weapon because virus-infected cells are secreting these and they're trying to warn other cells to be prepared. Interleukin amps up the immune response. It's like turning up the power, in order to get your B cells to secrete more antibodies, etc. Now, one strategy where the possibilities of the immune system is kind of unleashed and where they're doing research. When I made this presentation a while ago, I learned how to do this, so I'm obviously doing it for that reason. But is they will take cells and fuse them to an immortal cell like a cancer cell and get that cell to generate specific antibodies to target, let's say, a particular cancer. And not only that, remember how we talked about the base of the Y determines how it gets killed? They can generate antibodies that literally bring the chemical that you want, like a bomb. It's like escorting a bomb in and targeting that particular cell. So when you go and get chemotherapy now, it's putting those chemicals through your entire body, and the goal there is to take out the fastest growing cells, which are usually the cancer cells, but it also affects your other fastest growing cells. And that's why you can get really sick from getting chemotherapy. How much better would it be to escort the chemical you need just to the cells that need it, right? And just destroy those cells instead of harming all your other body cells. So that's some of the things they're looking at there. So can our immune system ever go bad? Yeah. We can have an immune response to things that shouldn't bother us, like pollen or nuts, peanut butter, those types of allergies or jewelry. If it's a really bad response, then you can go into anaphylactic shock. Your blood pressure can drop. It can cut off your trachea so that you can't breathe, and that can cause death. Probably what most of us instead will experience, hopefully, in our lifetime are more like a response. And these are memory T cells that amplify the response. If you've ever had poison ivy, is more East Coast. Here on the West Coast, have you ever had poison oak? Poison oak can be terrible, terrible response. Looks like this, oozy, itchy sores. And if you've ever been exposed to it, the next time you're exposed to it, you'll have a greater response and a greater response. Those of you who are allergic to like cheap metals, ladies or gentlemen that wear earrings, if you wear like cheap earrings, sometimes they make your ears itch. That's your T cells. My sister can wear a metal belt. Like, you know, belts don't need to be solid gold, right? And she can wear a metal belt, and her jeans can be between her belt and her skin. When she takes those off, you will see an imprint that looks exactly like the shape of the belt on her skin, even though her jeans are in between that, because it still triggers her T cell response, very sensitive to it. Just like nuts in a building can bother some people, even if they're in a package. Where can this cause problem? With tissue rejection. When we borrow an organ from somebody, then we can attack it. There are diseases where we attack ourselves, like lupus. Lupus oftentimes are limited to joint reactions where you attack the joints, but also it can attack your kidneys and just try to wipe your kidneys entirely out. It can attack the linings of your lungs and try to destroy your lungs when your own body can't determine self from non-self. And here it can attack your the sheath around your neurons, kind of like you don't have naked wires in your house, they have colored plastic coating. That's kind of what myelin sheaths do, and it attacks those, and then that causes problems in your nervous system. Okay, so let's get your notes on that, get that filled in. Cytokines as therapeutic agents, the word you're looking for in number 1 is signaling. In number 2, stimulates white blood cell formation or function. In number 4, the word you're looking for is warning. In number 5, the word you're looking for is prevent. In active immunity, it's exposed to an antigen. In passive immunity, number 1, the word is borrowed. Number 2, do not develop any memory cells. In allergies, immediate response can occur within seconds. 1C is life-threatening, drop in blood pressure. Delayed response is initiated by memory T cells. The response in B is regulated by cytokines. Blood type reactions, you attack any blood that you can build antibodies against. And in 13.5, immune system disorders and adverse reactions, the first one regulates when cytotoxic T cells or antibodies mistakenly attack, it's results when, mistakenly attack the body's own cells. And in the second box, causes unknown but sometimes appear following recovery from an infection. Recovery from an infection. Okay, now your digestive system, there aren't any things you need to memorize there, but there are a few words that you want to get. I'm happy to give you those words here at the end if you want to stick around for them. If you need to go, then you can feel free to, and that's it, sorry, in this lecture.